Great. So actually all I'm going to say today has already been said this morning. So I've really enjoyed all the talks and discussion this morning. They've been very wide-ranging and exactly what we were hoping to get from today. So thank you for that. But I was asked to talk for what I talk about when I talk about personalised medicine. And I'm going to give you a quick overview of that, except that's not right. Oh, okay. So as we've already heard, there are very different accounts of what happens when we talk about personalised medicine. I'm thrown because it's not up on the screen here, and I thought it would be, but let's see if I can do anything. I don't know. Yeah. I'll just look at the screen up here. So this is a Wikipedia definition and the NHS England definition. Personalised medicine is an emerging practice that uses an individual's genetic profile to guide prevention, diagnosis and treatment of disease, and it can help doctors select the proper medication or therapy. These are all very familiar to you, I'm sure, these definitions. And the NHS England website talks about personalised medicine and the 4 Ps, one of which is participatory medicine, and we've talked a bit about that this morning, about how we need to look beyond the technical. Genome UK, a government report, really listing that, or aspiring to the future of healthcare being all about genomics. But of course, the flip side of that is that medicine has always made attempts to personalise. So personalised medicine is, in and of itself, nothing new. People have recognised that, called it precision medicine and targeted medicine. But all these terms are, I think, not necessarily new terms. What's new is that developments in technology can help refine the granularity of investigations, diagnoses and treatments, but these may not necessarily result in better personalisation for all. And we've had some very nice examples of that this morning. So when I talk about personalised medicine, I try and talk about this broader construction of personalised medicine. And in fact, I think that sometimes when we talk about personalised medicine, it can become depersonalised, because we can end up talking about samples going in and patients being consented and the data coming out, and it's portrayed in nice glossy triple helix pictures that are shiny and blue, and with very sort of technical stuff inside a black box there. And so actually the person has been taken very much out of that. And I think what we've been talking about today, and what I'm really interested in, is looking at the other attributes that we need to incorporate in to really make medicine personalised. And these are not comprehensive, there's just some of them here, but context we've heard a lot about, that really to be able to personalise, we need to know about the context of the individual in front of us. Sustainability we've heard about, and then about the health service being trustworthy, developing relationships, thinking about diversity, and a sort of iterative relationship between the health service and research, and perhaps the commercial sector also, is really key to personalising medicine. But I think this sits in a society where we have very optimistic expectations often of personalised medicine. And I love this picture because it's what, on the left, sorry, because it's what people often expect, that as soon as we have a whole genome sequence, we'll be able to hold up a magnifying glass to it and know what's going to happen to us in the future, what our future holds for us. And that, moreover, it will give very clear predictions rather than stratify a population and still over-diagnose. I think William highlighted in his talk, and several others, about you might be seen to be at high risk, but that still then contains a lot of people who don't go on to develop the disease in question. But of course, this sits in particularly in a commercial sector, public-facing, sort of, it's very present on the internet and social media, that finding out about your genetic information will give you some sort of power, because knowledge is power. And I really like to ask the question about, when is this jumble of G's, A's and T's and C's, when is that actually knowledge and when is it just a confusing jumble? But I think there is a very dominant discourse that assumes that once we have our whole genetic sequence, we'll be empowered and know more about our futures. But just highlighting some of the work of Rachel Horton in particular, but others also in my group, that personalising often takes time and it's often much more uncertain than we might like to think it is. And that's because if you're looking at a whole genome sequence, we're trawling through that entire genome or genetic code, and we're evaluating the thousands of variants that we will find in there, including the thousands of rare variants that previously we had thought would automatically tell us about disease. And that, I think, exposes our ignorance about what much of this variation actually means, with what certainty. We talked about penetrance earlier, over what time scale. Is it 20 years in the future? Is it now? And in what particular clinical context? And a clinical picture, so signs and symptoms or family history, can really help exclude many variants. But that is much harder if we're trying to do screening approaches where we haven't got that clinical context to apply to that exclusion process. So that complex and exploratory nature and ability to see other information means that research and clinical practice become entangled in a way that we're not necessarily comfortable with, and it's all much more bouncing between different areas in order to personalise. I think we should really embrace the fact that uncertainty is not a failure, but it is a difficulty if it's not addressed openly and honestly in the face of these often very deterministic discourses about genetics. I don't know if people saw this article earlier in the year from Christina Pagel about screening, and she, I think, voiced a very common view, which is that more cancer screening sounds like good news, but can the NHS cope with them? So that second bit, I think, is a really good question, but actually our article went on to say that if you screen, you're going to be following up an awful lot more people who are initial screen positive but don't go on to develop the disease. And how do we balance those our resources, but also how do we balance the expectations of people that screening will give them a yes/no answer, when actually usually it gives you a maybe and puts you in a sort of sequence of events that can be costly, timely and anxiety provoking. And I just put this up, this is one of Rachel Horton's cartoons where she talked about newborn screening, and actually what that's trying to show you is that this baby is tested positive for a genetic condition at birth and is then followed up on the NHS. It's a heart condition that the baby is found to have, followed up over time, but over 20 years, but never actually develops the condition in question. And the labelling of someone in that setting, we just haven't got our heads around fully as we're talking about newborn screening through whole genome sequencing. I'm going to go over this very quickly because Fahrenheit did a beautiful talk about diversity in genomics, but the message is, if you haven't already got it loud and clear, that despite many calls to diversify genomics, actually things are getting worse at the moment, that are the data that we're looking at is becoming more and more skewed towards people of Northern European ancestry, and we really need to do something urgently about that to make sure that this personalisation through genomics is equitable globally. And another way of thinking about that is also if you look at how the world's population has increased, and that's projecting we'll be at 10 billion people in the year 2058. That exponential increase, I think, highlights beautifully how difficult it will be to personalise medicine for all, because you'll be able to personalise for a few people in that massive global population, but as the population increases and increases, what we need to be thinking about at the same time is making our approaches equitable and asking the question, when we personalise, how much are we depersonalising for others? And this cartoon, I think, sums up very nicely what people often think about the NHS, the health service in this country, that it's too busy and too sort of stuck in its old ways to accept the idea of a round wheel rather than a square wheel. But I think that sort of negative construction of the structural problems in place for personalising medicine is not always helpful. We need to be thinking about what actually is required to take some of these fantastic developments in genomics and make them work in a health service and in society. So that's my last slide, that I think we can all agree that scientific and technological advances promise massive improvements in personalising medicine. That's not up for debate. And that genomic enthusiasm is really welcome, but we also need to focus on making the future of genomic medicine realistic. And we need to also recognise that we shouldn't always be talking about the future. We need to be thinking about how we're implementing some of those things now and how we can better implement some of those things now. Emphasising how genomic variation contributes to disease, often much more complicated than people think or that headlines might suggest, and that we urgently need interdisciplinary research, policy and public dialogue and engagement to understand and respond to these advances effectively. And that's why I'm so pleased we've got a day like today where we're doing exactly that. So that's what I think about.
Thanks, Andy. Great. So that's a great start. And I want to hand over to our second speaker, who's Julia Frost, who's Associate Professor in Health Services Research in Exeter.
Thank you.
You haven't got slides, have you? So
no, I want to. Yeah, that's great. The Slido. To think about what's on that and what I'm going to say. So when I talk about personalised medicine, I talk about drug development processes and democracies. And in this talk, I'm going to talk about 3 different types of democracies. And I talk about it in this way because I'm currently seconded to a biotechnology company that makes drugs for patients with rare diseases or orphan diseases. And I've been observing how this company interacts with the patient organisations it works with. And by that, I'm really clear that I mean non-for-profit organisations that focus on the needs of the patients with rare diseases and their carers. And as a counterpoint to that, I've also been interviewing a bunch of patients and the healthcare professionals that look after them in the southwest of England. And this work came about because the physician who looks after them told me that they can't get into clinical trials and they're having real difficulty in accessing the different trials for potential new treatments and indications that would help them. So it's taking up that challenge and then looking at it as a counterpoint to their patient organisations. The work's funded by the Medical Research Council. I'm working with Catherine Pope, and everything that I do has a patient advisory group that tells me all the things that go in and poke around in pharma and ask them questions there. So quite a different model of research. So as some of you know, the challenge of Health Research Authority ethics, while I was waiting for that, I did a big systematic review of the literature about how patients get involved. And it's really interesting. So it's a very pharma-facing literature to start with. But we found 13 papers that we included that were either qualitative studies or patient, well, they're kind of masquerading as PPI, patient public involvement, where the patients are being interviewed by their own physicians about their needs. And it seems really interesting to us that the only way that you get in those kind of academic medicine papers is if you have an established relationship with the physician, or you attend scientific symposia or meetings. So these aren't everyday patients, is what I would suggest. And then through poking around in pharma, and I'm going to keep referring to it as that, I found all these market access reports. So this is something that pharma pays for, but they don't do it themselves. They do it through third-party organisations, vendor organisations, and they collect, this is called patient intelligence or the patient voice. So when I looked at this literature that the pharmaceutical company have allowed me to access, we see that the only way that patients get into the market access reports is if they're a patient organisation with an established relationship with one of these third-party organisations, and if they can respond within the 4-week window of the pharma finding an indication and wanting to do their early phase trials. So again, I want to suggest that this is a particular group of patients who gets into this patient voice exercise. And I want to think about this as the first form of democracy, as aggregative democracy. So this is how patients get counted or pulled together into what's often thought of as an average patient, and it's reported as an average patient. Their data all gets aggregated. And it's a really interesting idea to think about an average rare disease patient when probably they aren't very average at all. And then another way that patients get aggregated is in the umbrella patient organisations. And these are meta-patient organisations. So something like the European Patients Forum is a coalition of over 74 separate patient organisations. And this is really interesting when we're thinking about personalised medicine, which I talk about as individualised or tailored medicine, that we're given an averaged or mean experience. And we're not really sure how this happens. But it's something about collectivisation. If we think of this as political science, the collectivisation is how you make things manifest and realise there's a strength in patients coming together. There needs to be enough patients to kind of lobby a group to behave in a particular way and to make a business case for the new indication. We talk about unmet need, patient unmet need, but it's interesting who constructs that. So what I've been doing is thinking about other ways that we can think about democracy, and I've been doing that through ethnographic methods. So one of the things that I've just been observing over in Europe is how this particular pharmaceutical company works with patient organisations in a particular way around a patient advisory council, is what they call it. So as an advisory council, you can imagine it's a consultative body, and it's the idea is that it better aligns the company's activities around the unmet needs. So I've been watching their meetings, their asynchronous, their face-to-face meetings, and they have asynchronous platforms that patient organisations can comment on as well. So I've been looking at the kind of data that goes into them. And what I think this is another type of democracy. This is participatory democracy. And by that I mean the direct participation of citizens in the regulation of key institutions of democracy. At face value, participatory democracy sounds like a good idea when we're talking about participation. But on the other hand, it's about patients being given the opportunity by the institution on the institution's terms as a consultant. The agenda isn't being set by the participants themselves in any way, and it risks being reactive, reductive and vague. Because what I've witnessed is it actively avoids any risk of confrontation, by which I mean they talk about soft topics which are purposefully chosen to avoid any confrontation. So when the agenda is being set, it's a very safe agenda that's set. And it does feel like a bit of an echo chamber where they talk about things like how can they facilitate a patient organisation reflecting back what the pharmaceutical company's just decided. So the interesting things to think about. What's interesting is the patient organisation representatives want something other than the current offer of consultation. They don't want a set of guidance or policy that the pharmaceutical company wants. They want to have the company be accountable, report quarterly on the impact of any involvement that they have. So over the last 2 years, what I've been witnessing is that they want to set rather than respond to the agenda. And in some, they want to influence rather than want to approve pharmaceutical practice, which I'll come back to. As I said at the beginning, concurrently with that, I've been interviewing patients and healthcare professionals in the Southwest. And they really have been struggling to get into clinical trials. And the way that I've recruited them is through clinicians' lists. So these may be patients who did take part in trials, or some took part in many and some took part in none. So they're quite an interesting demographic. What's interesting from those interviews is that these patients can lose stake in their drug pipeline at any point. So access can be denied because they live too far away, we're down the pointy bit of the UK, and they've got to travel 250 miles to get to a tertiary centre to take part in a trial. They don't meet stringent criteria, and we've already talked about the impact of comorbidities, which you will have if you're an older person. So, you know, a lot of these people don't meet the criteria. They don't want to take part in anything that's going to give them a placebo. If they've got a rare disease, they see it as a terminal diagnosis, they want to have an active treatment, they don't want to risk carrying nothing. They drop out because of side effects and no sense of improvement, even though the purpose of the drug might be to limit or inhibit disease progression rather than be curative. And it's particularly upsetting for patients when trials get stopped, when they've really invested in them, and they don't think that they're fully prepared for something which is quite a common outcome in a phase 3 trial. And the other thing is that these patients, these more naive patients, don't recognise themselves in patient organisations at all. They see them as being overtly cheerful, with their patient organisations all wanting a cure and believing that a cure is coming, or they think they're morbid. They don't want to sign up to anything where people talk about their angel wings. They find this place is really depressing, and it's also they see their future selves and they don't want to go anywhere near it. So again, we can think about what that means for these patients being outside. Conversely, there's also another group of patients who have sufficient capital, and these patients just go private. And that was really quite interesting to me, the number of them that can just dial up a hospital in London and circumvent any kind of trial science whatsoever. Or they can buy their orphan drugs off the internet. So there's something the critics have argued that participatory democracy risks perpetuating inequalities, and I think most of the talks this morning have gone there with inequalities. And it's something about the most at-risk patients just can't get into trials at all, whereas the most advantaged patients just circumvent the whole process. So when we're talking about the patient in trial science, we're talking about very particular patients. And one of the speakers around political theory said about 30 years ago, individuals are likely to find decision-making so burdensome and inefficient, most will withdraw to cynical apathy. They will leave most decisions to an activist few who will ironically make decisions based on the authority they derive from a participatory process. And I think that sums up really quite nicely what we're seeing. In contrast, we can think about deliberative democracy. And this means any practice of democracy that gives deliberation a certain place. Political theorists broadly agree that deliberation is free, equal, open-minded communication that leads to civil engagement, activity, empowerment, efficacy. And I think that's what a lot of the patient organisations are after, really. But one of the challenges about how we do something different is the regulatory oversight. So as many of you know, the Council for Harmonisation of Technical Requirements for Pharmaceuticals for Use in Humans, or the ICH, thinks that it's not okay for pharmaceutical companies to talk to patients directly because they fear marketing or lobbying. So it means that they're only ever going to talk to a particular pool of patient organisation representatives. We've argued that the current regulatory framework means that it prioritises the patient voice upstream in the drug development life cycle, which further renders the perspective of the downstream patients even more invisible than it was before. So the problem is that the ICH knows this is a challenge and it's issuing guidance for potential future recommendations. But nobody that I've spoken to in the last 3 years knows how to do this. They see that there's a need for fit-for-purpose regulatory practices, but we don't know what it looks like. So one way that we can think about this is perhaps they could employ deliberative methods. So one of those deliberative approaches could be something that's called invited activism. So this is what you would use for people who don't engage in acts of spontaneous activism. So we think about climate protesters. So somehow we need to work with them in a way that better engages them. So what the patients are suggesting is that pharma needs to reach out to patients and work with them in the spaces that they inhabit. Really novel idea. So maybe in the local places that patients inhabit, so where they get checked up when they're not going to the tertiary centres, in their GP practice or their community hospital. So they think this would be the place where they could put their validity claims to pharma and challenge the current drug development processes that construct in-patient voice. Another way is thinking about political translating, and that refers to practices of inclusion initiated or chosen by marginalised populations. And the way that we're going to do this next is through a citizens' jury, which I know has been done here in Oxford. And one of the key witnesses in this is going to be our patient advisory group, who is going to be talking about their role in this process with a naive group of patients with a rare disease, as well as some people from the pharmaceutical company and clinicians who treat them. And we think the witness statements will be presented to rare disease patients and naive patients who will act as the jury, which will provide, we don't think will provide a solution to patient engagement, but it will we hope it will provide a process for deliberation that has the potential to be replicated everywhere else. So the point I'm making is when I talk about personalised medicine, I talk about new processes of patient-public deliberation to better inform drug development.
Thanks very much, Julia. The final speaker is Professor Julia Knight, who's a Professor of Genomic Medicine here in Oxford and Deputy Director of the Wellcome Centre for Genetics.
Great. Thanks very much, and it's a pleasure to be here. Really grateful for the opportunity. I'm going to tell you a little bit about what I talk about when I talk about personalised medicine. I just wanted to, first of all, expand a little bit on one of the points that Annika was making, which is when we think about human genetics and genomics, we're largely driven by technology and our ability to generate information. And so when I think about personalised medicine, it's really thinking about genomics in a very holistic way and thinking about the different steps that we can now measure in moving from DNA sequence through RNA and protein and then different cellular phenotypes. So all the different omics that are now possible to look at, whether it's transcriptomics or proteomics, would be part of my kind of thinking when trying to develop personalised medicine or teach about it or talk about it. So thinking about the different settings in which I might be doing that, I begin with thinking about education, because if we're going to implement any of these new advances and opportunities in personalised medicine, there needs to be a workforce to deliver that, and there needs to be the innovators who are going to continue to develop things. So, for example, we've recently set up an MSc in genomic medicine here in Oxford that takes advantage of the very rich research infrastructure. But if I was talking to the students there, I'd probably go for some really well characterised examples, and I might talk about cancer and how we can use knowledge in particular mutations to target therapies. Or I might talk about pharmacogenomics, recognising that we've got relatively few examples at the moment, but we do, and we know that if you're going to be put on warfarin, we'd use some of your genetic variation in particular enzymes, or indeed if you're going to be put on an antiretroviral drug for HIV, such as abacavir, we'd use knowledge of your underlying genetics to guide treatment. And I'd say to the students that we have an opportunity to use personalised medicine to move away from our current clinical syndromic definitions of disease to more precision. Because we can use a syndrome, but we recognise that there's a whole series of subgroups within that, and those subgroups will almost certainly have different mechanisms, and they will have different therapies that will benefit those groups. And currently, by treating everyone within a clinical syndrome, we usually get away with it, but we are causing harm undoubtedly in a small proportion of those patients, or at least they're not deriving benefit. And so greater precision in how we reduce therapy. I'm fortunate to be able to run a research group, and if I was thinking about our current research, the clinical syndrome we'd really like to move on from is sepsis. So this is a label that gets given to patients when they have an inappropriate or maladaptive immune response to infection. And this occurs in only a small minority of people, so lots of you might develop pneumonia, but only, hopefully none of you, but a very small proportion of people will end up in hospital or on intensive care. And in that situation, your immune response is actually causing the damage. And we have been using transcriptomics as a way of measuring expression of all different genes, and a way then to say in an unbiased way, let's cluster these patients based on that signal. And it turns out that that clustering is interesting, and it takes you to new groups who have particular problems. For example, we've got a group of patients who've got an issue with their neutrophils, we think, where targeted therapy would be much more helpful. But if I'm having those conversations with the research group, it'll be at a very technical level about these different technologies and how we can most effectively apply them in different patient cohorts. And then a few weeks ago, we were able to have a different conversation, which was with a diagnostic company thinking about personalised medicine. And there, we wanted to take this discovery to say, actually, how could that move to a point-of-care test? So could we measure particular RNA biomarkers and do that in a way that's rapid enough for these very sick patients to get an answer which doesn't rely on RNA sequencing, but will rely on a PCR-based method. And if you're talking about that in the setting of a company, it's a different conversation. It's very much about the practicalities of how a test might work. Can we do it in 30 minutes? What can we push in terms of different parts of that technology? What platforms already exist? And how are we then actually going to use that in practice in a clinical situation? And what are the clinical use cases that we can really precisely define that doctors will then say, in this setting, we're going to use that test? And then there's the question of how you more broadly set that in a healthcare setting. And so I work with Padraig and other colleagues in the new NHS Genomic Medicine Service. And that's about establishing the clinical pathways whereby this sort of genetic and genomic knowledge can be rolled out into both diagnostics and clinical care. And there you're having to think about the constraints with which the NHS is currently operating. Will there be the opportunity in terms of the infrastructure, the current laboratory setups that exist, the ways that people can actually get the necessary financial case for using a particular intervention, and indeed, coming back to this workforce question, who's actually going to be implementing that test, and can you demonstrate the health economics of that? Can you demonstrate that what your test when it's implemented, will be equitable in terms of different population groups that you might be using it in. So that's a different conversation. It's helped by the fact that my background includes working in the healthcare system. But I'd say for me, it's a really broad spectrum of different people having these conversations with, who've got their own priorities. But we need to remember that ultimately what we're trying to do with personalised medicine is to maximise benefit for the patient and minimise harm. And if we can get that right, genetics and genomics is one of a number of different ways of getting to that personalised agenda. And indeed, it might be that in 5 years' time, we're thinking about how machine learning and other approaches allows us to take all sorts of clinical variables and other variables, as well as these more molecular ones, to get us to a really precise diagnostic point and opportunity to identify new therapies and implement them.
We have to bring the chairs nearer to the microphone. I just wondered, first of all, just before I open up, whether any of you, kind of reflecting on what other people have said, whether you're having any initial thoughts. I mean, you went first, Annika, so maybe you've had a chance to listen to other people's thoughts after yours.
So I think that where I'm sort of stuck, I suppose, in thinking about the journey is that the expectations of this very technical beast of genetics, genomics, but other omics as well, the view of this scientific development is over-optimistic. And I don't know how we rebalance that, because I think if people have over-optimistic or very optimistic expectations of it, then it's much harder to meet those, and it's much harder also to say to people at the beginning of their journey, well, there might be downsides to this as well. It might not give you the picture that you were hoping for, or you know, in your particular case, you don't come up with a diagnosis despite having had your whole genome sequenced and all your omics done and everything like that. So it's about having a realistic conversation, I think, with people that sees the technology and the massive advances in technology in the round in a sort of society in which we live that isn't fair and isn't as nice and clear cut as a lot of people expect. So I think what I'm struck by is that we'll probably all agree somewhere along the line, but that it's very hard. Certainly I'm thinking, you know, as a clinician, I'm thinking of the conversations that I have with patients where I'm sort of saying, well, it's not going to be as clear cut as you think it's going to be, and how we can build that in more effectively into those journeys.
So the problem people, as it were, the problem discourse is about making it look easier than it really is, and making it more likely to be successful than it really is.
Maybe, but that happens in all walks of life. Yeah. So, you know,
HS2 is a good example.
Yes, exactly. And you know, any headline will make the article below it sound nice and straightforward. So it's not an unusual problem, but it persists nevertheless.
Julie, I just wanted to ask you to just say a little bit. So the end, your talk was really, really interesting, outlining these different kind of political processes and ways to think about it. And I just wondered if you could say, in the light of the things you've heard, say a little bit more about the sort of, as it were, the next step. How might that lead to a better way of, or better, it might not be the right word, more appropriate and constructive way of thinking about?
It's difficult.
Yeah.
So my background's actually in ITU, and I think it's the same thing. You've got the techno-rational shiny, but it's relational. So I've just been on a lab tour and I've seen the techno-rational shiny new thing that's going to be really helpful in the global north. And the patients are saying primary endpoint's fatigue. The shiny doesn't talk to the primary endpoint that the patients are talking about. So yeah, so I do think it's dialogue and having patients in the room to be listened to and set the agenda.
Right, Julie, I thought you were, although you weren't, but it wasn't the shiny kind of personalised medicine thing, I thought your journey, as described, even if it was in the first instance for a limited number of settings, whether it's sepsis or the other things you talked about, it seemed more hopeful. I mean, you seem to be more, you know, we're moving in the right direction. Let's not get too excited about it, but nonetheless, there is real potential here. Is that what you?
Yeah, I genuinely think that. I think it's certainly taken much longer than I kind of naively expected in going into this, and I think it's really important to recognise, as Annika was saying, that if you talk to physicians, they would say, well, we do personalised medicine, and so we don't. It's very important not to alienate that much broader constituency and show how we're adding value and tools to help with that process, rather than reinventing something or coming in with a new concept. We're certainly coming in with new concepts in terms of the ways that we can do this, but the overall approach is something that we're building on. And I think that, as you were highlighting so nicely, the need to not only be involving that more clinical community, but the patient community in that, so that we can get good representation and we can prioritise appropriately. But I'm optimistic, but we certainly cannot overpromise. And if you think back to when the Human Genome Project was announced, and then the initial sequence, people expected for much faster return.
That's a problem throughout healthcare, isn't it? That research findings take something like, on average, somebody will correct me, 25 years to be implemented into practice. And we all assumed that genetics and genomics were going to change that, because once we had our genetic code, it would go into practice straight away. But it's no different to any other research finding. It takes a long, long time to implement into practice.
You do sometimes get things that make a big difference. So thinking about vaccines, for example, the fact that we were able to develop vaccines very quickly to COVID was because of new technologies and new scientific developments, which were in place as kind of platform technologies. So it's not necessarily that things there are examples of rapid progress.
Yeah.
But the challenge is talking to pharma, well, the patient organisations that work with pharma, is that because of COVID and austerity, they're now even more financially dependent on pharmaceutical companies and have less of a voice to argue against any new shiny things as well.
My reflections about the talks have been, I think that my priorities, I don't know if I've got the attention span to remember them all, but the first one I think we didn't explore as much, but I think is really important to think about, which is the sort of changing boundaries of healthcare, that point that you nicely raised about how healthcare data is coming in different angles, mobile phones, whatever, direct-to-consumer tests. We need to really be thinking about how that is influencing the personalisation of medicine. And then I think that the sort of equity factor is a really important one that we need to get in there, that is under-recognised at the moment, and it's particularly, I would say, under-recognised in genomics, because there is an assumption that the genetic code is uniform, and we don't recognise, as Faranak highlighted very well, that because our understanding of people from the global south or from non-European ancestry specifically is much more limited, that we are widening inequities by not addressing that problem. So we really need to focus on that problem, I would say. And then I think the third big thing that came out today for me is the idea of that, it might sound, as Sarah, you said, that sustainability, how does that relate to personalised medicine? But it does in a massive way, because we've got this exploding global population, and we've got these widening disparities. So if we're focusing all our attempts to personalise on an already affluent and privileged group, then we're really doing the world a disservice, and that we need to be paying attention to how our attempts to personalise are having not just a carbon footprint, but an environmental footprint that's going to make things worse for the vast majority of the population, rather than the small minority of the population that is benefiting from them. So they would be the three. I'm sure there were lots more messages that came out of that, but they're my three take-home messages from today. And I just wanted to thank everyone. For me, it's been such an engaging day, and really interesting discussions. And Susie and Kate in particular for organising this. You just got on with it and did it. And then of course Kat and Thea for making it all smooth and really a very enjoyable day. So thank you all very much for participating.